My name is Claire Gerard, and this is my video presentation for Field Experiment 2. Today, I'll walk you through my study on the relationship between fucus cover and nudibranch abundance in tide pools along the rocky coast of Nova Scotia. First, I'd like to make a land acknowledgement. I conducted this research on the coastline of Mi'kmaq, the ancestral and unceded territory of the Mi'kmaq people. I am grateful for the opportunity to live, learn, and conduct research on these lands and waters, which have long been home to deep ecological knowledge. Tide pools are dynamic intertidal habitats, offering refuge for species like nudibranchs, small shellless sea mollusks that are often sensitive to environmental structure and vegetation. One major habitat-forming species in our intertidal systems is Fucus vesiculosus, or bladderwrack. I wanted to explore whether the abundance of nudibranchs varies with the amount of fucus present. Here, I have a couple of examples of nudibranchs that we can find off our coast here in Nova Scotia. In fact, these are photos I took during the data collection of this experiment. My research question was, does fucus cover influence the abundance of nudibranchs in tide pools? I hypothesized that nudibranchs would be more abundant in pools with moderate to high fucus cover due to increased habitat complexity and potential prey availability. And here I have an example of what exactly does fucus look like, also known as bladderwrack in this image on the right. My fieldwork was conducted at Peggy's Cove, a rocky intertidal site on Nova Scotia's south shore. Sampling was completed on May 22nd during low tide, ensuring that all pools were accessible and visible. Here, I'll explain a little bit more about what exactly I did for my field methods. I selected 20 distinct tide pools, each isolated enough to be treated as an independent unit. For each pool, I visually estimated the percent cover of fucus using a 0 to 100% scale, and then categorized each pool into one of four cover categories: absent, 0%, low, less than 25%, moderate, which is 25 to 75%, or high, which is over 75%. And here I have an example of three of those categories. I searched each pool for nudibranchs for a fixed period of 2 to 3 minutes, recording the number of individuals observed, along with species ID when possible. I also recorded basic environmental data such as depth, substrate, temperature, and GPS coordinates, using a field datasheet and a thermometer. Pools were selected across a variety of tidal heights and exposures to reflect the natural heterogeneity of the intertidal zone. After fieldwork, I analyzed my data using RStudio. I created box plots, ran an ANOVA test to test for differences in nudibranch abundance among fucus categories. And I also used a Kruskal-Wallis test and a Wilcoxon pairwise comparison to confirm the trends. The results showed a significant effect of fucus cover on nudibranch abundance. My ANOVA test returned a p-value of 0.0015, and my Kruskal-Wallis test confirmed the significance with a p-value of 0.0028. Post hoc tests showed that nudibranchs were significantly more abundant in pools with high and moderate fucus cover compared to pools with no fucus at all. These trends are further reflected in this box plot here, which shows again high and moderate levels of fucus tended to yield a higher nudibranch count. These findings align with established ecological theory and previous research. For example, Wickstrom and Kotsky, 2007, found that the presence of canopy-forming macroalgae like Fucus vesiculosus significantly increased invertebrate richness and abundance, while their absence led to reduced community structure. Similarly, Umanzor et al. In 2017 showed that intertidal macroalgal canopies enhance macroinvertebrate distribution by offering thermal protection, moisture retention, and physical refuge. Yet, specific studies on nudibranch habitat associations in Nova Scotia remain scarce, making this study a valuable foundation for future research into their ecology in local intertidal systems. Interestingly, I observed 4 species of nudibranchs, including Chorophella and Dendronotus species, all found exclusively in vegetated pools. This could support the idea that vegetation influences not only nudibranch abundance, but also species diversity within these pools. Ecologically, this seems to make sense. Fucus provides shelter, surface area for feeding, and a temperature buffering. If nudibranchs are visual predators, this may also benefit and provide camouflage or protection in vegetated pools. This supports the idea that habitat structure drives species abundance. While these findings are definitely exciting, my study certainly had its limitations. For one, it had a small sample size, as there was only 5 pools per category. A second limitation is that there was only a single sampling time, which was at midday, low tide. And the third, there certainly is the possibility for observer bias. I have gone tide pooling in the past in Nova Scotia, and I have found nudibranchs in the past, but it is certainly a possibility that I am only attuned to what I had seen prior. Despite these limitations, the patterns were consistent and statistically significant. Incorporating two-eyed seeing, I recognize that while my work uses Western scientific tools, it can be deepened by Indigenous knowledge. For example, Mi'kmaq traditional knowledge views intertidal habitats as interconnected systems and not just species-specific niches. Future work could possibly track seasonal cycles of algae and nudibranchs. It could maybe examine cultural uses or observations of invertebrate abundance, or it could include co-design studies with Indigenous communities. Understanding biodiversity from both perspectives leads to a more holistic conservation approach. In conclusion, I found that nudibranch abundance increases with fucus cover, supporting the idea that structural complexity promotes biodiversity. This experiment highlights the importance of habitat-forming species in shaping tide pool communities. Outside of the purpose and goal of my experiment, I do find that this is very valuable just for the sake of proving that there is lots of life to be found in our Nova Scotian waters. There is a misconception that there isn't much to be found and there's not too much life within our waters, but I do believe that this experiment was a great proof that there are amazing little things to be found if you have the patience to do so. If you have the patience, our waters will absolutely reward you. In addition to my experiment, I wanted to provide a little tutorial of how I was able to find nudibranchs within Fucus. I certainly don't expect people to replicate my experiment, but if people are tide pooling or close to the water and they want to be able to see if they can see nudibranchs, I believe this could be helpful. So here I can be seen moving the bladder rack back and forth and kind of just fingering my way through and being very, very gentle while I do so. A lot of nudibranch species are very small, some smaller than others. Luckily enough, a lot of them are brightly colored. So you should be able to catch a glimpse of one if you are gentle and patient enough to do so. I promise you, you will absolutely be rewarded with patience and gentleness. I really hope you enjoyed this experiment, and I really hope those of you that watched this are able to go out and look for nudibranchs on your own.